Welcome to the video of the tutorial for Pseudobulk analysis using tools like Decoupler and EdgeR. My name is Diana and I will do the hands-on part of this tutorial in Galaxy together with you in this video so that it is easier to follow when you are self-learning at home. Now, the first thing we're going to do, which is easier to follow, is we're going to click on this hat and we're going to go into the Galaxy Training Network site. By clicking the hat, we directly come here to this Galaxy Training Network, and you will have to find the tutorial, Pseudobulk Analysis with Decoupler and EdgeR. Go into the single cell topic and then look for the Pseudobulk analysis tutorial. There's a few things that we will address today, and by performing this analysis, this tutorial, you will understand what Pseudobulk data is, what is the power of Pseudobulk analysis after single cell when you want to perform differential expression. And you will learn, of course, how to generate the Pseudobulk count from single cell data and how to use the bulk RNA-seq analysis tools for differential expression after aggregating the count from single cell into Pseudobulk. Now there's further information. You have here datasets, workflows from this tutorial, the answer history, which is very useful if you want to see all the steps already performed. They are all here, and you can click on them and it will take you to the answer history. Now you can read about Pseudobulk and the details of it and the power and the positive aspects of performing Pseudobulk instead of doing a bulk experiment or just performing single cell differential expression analysis with the single cell approach. What we will do is we will directly go into the get the data section of the tutorial, and here it details a little bit about what kind of data we will be using today. So the data format is an nData object. This is the type of data that you need for this tutorial, an nData object. And our nData include immune cells. In this case, we have non-classical monocytes and plasmacytoid dendritic cells from two types of tissues, bone marrow and peripheral blood samples from humans. Now, we are going to perform an analysis to see if non-classical monocytes and dendritic cells express different genes in bone marrow compared to blood. So that is the focus of the Pseudobulk, and in this tutorial we assume that you are familiar with this data formats of nData, Surat, and that you know how to annotate clusters that you have used and have already prepared your nData by analyzing it first with ScanPy or Surat. And now you want to calculate the differential expression in a more statistically powerful way, which is why you go into Pseudobulk. Now our data object, the one that we will import in Galaxy, have already our cells annotated. So we have identified which cells from our datasets are non-classical monocytes and which ones are plasmacytoid dendritic cells. And these are under the annotated label. Also, we have condition, or sample group, because this data comes as well from patients with cardiovascular diseases. So some patients had myocardial infarctions, others not. This is not relevant to our analysis because we're comparing tissues, but you also have this information in the metadata of your nData object. You also have the batch, so the sample identifier, and you also have the tissue, so which cells correspond to bone marrow and which ones to blood. Now, we're going to import our data. We're going to copy this link from Zenodo. We're going to upload, paste, fetch data, start, and we let our data upload. Great, our data is there. So now the next step is to inspect the data. So we're going to click here and it will take us to the Inspect Data tool. And we just want general information about our data object. Perfect, we have our results now. We're going into Inspect Data, and to take a better look at this results, we will click on the eye. And this will show all the observations and variables that we have, the metadata that we have in our data object. In this case, the data object has n numbers of observations times n numbers of variables. So this means that it has 9,118 cells and 18,926 genes expressed by all those cells. Now we can see here as well all the other metadata and annotations that we have in this data, tissue and tissue group by condition as well, and so on. And yeah, here you can see all the layers. Importantly, we have here our layer that it's called raw counts, because when you're performing Pseudobulk, you cannot use normalized counts, which normally you will get after a single cell downstream analysis. You need a raw count. So at some point during this single cell downstream analysis, you should have frozen your raw counts somewhere in the data in a layer that you will call raw counts or so on. So great. Now we go back and we inspect the general information. Here we have a couple of questions. How many cells and genes are there in the data, which we already answered? And what other information can be retrieved by this tool? So the Inspect Data tool can retrieve more than just general information. It will all depend in the parameter that you choose. You can go deep into inspecting observations, variables, and so on. Okay, so the next step is the generation of the Pseudobulk count matrix with the coupler. So at the moment, we have our data and we have cells that represent a data set. For each cell, we know whether it expresses one of those 18,000 genes or not. But we don't want to know by cell the expression. Instead, we want to know by sample. So we want to group our Pseudobulk counts by sample and also by tissue. So to start our Pseudobulk analysis, we need to make sure that we have the metadata of the annotated cell types that we have, for example, condition, disease, batch, which represents samples, individual samples, and so on. And as I mentioned earlier, there should be a layer containing raw gene expression counts. If you don't have this raw counts, there's another tool in Galaxy that is called Data Operations, and will allow you to copy, for example, the matrix of your single cell data as a raw counts into your data in a new layer. But you have to be a little bit careful with this copying, so it has to be done correctly. You have to make sure that from your previous single cell data that you're actually copying the raw counts. Okay. Okay, so the next step is to generate the pseudobulk count with our decoupler tool. Now we're going to click here, and this will take us to the decoupler pseudobulk tool, and we will pick our data. Now it says produce a list of genes to filter. No, we will leave the observations fields to merge empty, and let me fill out this information now. We know that they are grouped by annotations, so it's the column containing cell type annotations. Annotated is the name, and batch is for each sample. The layer is called raw count, the one that contains our raw counts. The factor fields, which will be used in the decoupler tool later on, in our case is tissue, because this is an experimental variable that we want to compare when we input our data into the EdgeR tool later. We want to know for each sample what is the differential expression in non-classical monocytes when they are in bone marrow compared to blood. Now we say use raw count, use raw no, because sometimes there's a layer called a raw, and this has the raw counts, but in our case our layer contains raw counts, is the one that has these counts. Then we will minimum cells put 10, produce plots, yes. We will not produce data because we really don't need it. Then minimum counts 10, minimum total counts 1000. Minimum counts per gene per contrast field 20. Enabling filtering by expression, yes. And this is just the size of the plot. We're going to make it a bit bigger. And that's it. We're going to hit run tool. And now we wait. Great. This tool takes a while, so don't get discouraged because Decoupler takes a while until it generates all your Pseudobulk files. But you can always read our PhD comics here. Okay, so now we have several outputs from the Decoupler tool. One is the count matrix in a tabular file. Let's take a look at this. It's kind of like a large file depending on how many samples you have and how many cells and so on. But we can clearly see here that in this case of the data, we now have our samples grouped together. So our counts are aggregated by a sample. So bone marrow, non-classical monocyte, bone marrow, non-classical monocytes, bone marrow, non-classical monocytes. And now we have blood. We have non-classical monocyte blood 1, blood 2, 3, and so on. So we have here the non-classical monocytes samples, and if we go further in the columns, we will find the dendritic cells samples from blood and also bone marrow. We have only 3 bone marrow samples and 7 blood samples per cell type. And in the column 1, you will see the gene names and how many times they are expressed in each sample. In the past, the column that you will have will be cell 1, 2, 3, and then you will have another column that will tell you whether that cell is coming from bone marrow, or like you will have a metadata annotation whether that cell is coming from bone marrow or blood. But in this case, we have now aggregates of these counts. So this file is very important in the count matrix and it's a tabular file. We also have the samples metadata, which is a factors file that will be useful to generate a contrast file for the EdgeR tool later. We have the genes metadata with the gene IDs, with the, well, a lot of information. We have another output with this, which is the genes metadata, which contains columns with the genes in metadata information, right? And we have plots that are interesting to take a look at, and it will tell you the log 10 of total sum of counts versus the log 10 number of cells in each sample. So this is each non-classical monocyte sample. It doesn't tell you whether it is from blood or bone marrow, but you can see here that the blue dots are the non-classical monocyte samples and the yellow dots the dendritic cell samples. Okay, now we also have this other plot, number of samples, log 10 sum of counts, and so on. And we have another file that is the file that tells you per the contrast that we chose, which is tissue, because we want to compare the differential expression of the genes in a specific cell type between tissues, whether you have to ignore certain genes or not, but we are not going to take a look at this file at all in the tutorial or use it. It's also empty, so yeah. Now here we have some questions. It's how many outputs does the decoupler tool generate? Well, we already saw how many. In total, I think there are 1, 2, 3, 4, 5, 6, 7. And it could be an 8th one if we would have chosen to generate the AND data of our pseudobulk counts, which was possible, but we didn't do it. It also explains here what the outputs represent and the interpretation of the plots, which we already skimmed through them. And the ones that from this outputs, which ones will be used in the EdgeR for differential expression analysis is, I think, an interesting question. So the Pseudobulk count matrix, which is this tabular file, is a primary input required for analysis using EdgeR. And then we also need the samples metadata and some other files that we will generate in the next steps. Now, then part 1 of sanitation steps, this is very important and it could be confusing, but let's do it bit by bit and we will be able to generate all the corresponding files for later. Now, we're going to click on replace text in this tool. And for this tool, we will need the count matrix and we're gonna clean it. So we're gonna ask the tool to find this pattern and to replace it with an underscore. So in the count matrix, please find this pattern and replace it with this. And then we click Run Tool. So the other step that we will do is we're going to remove some columns from the genes metadata file output. And we are going to remove the columns that starts, well, that has a header that starts with the name start, the other one end, and the other one with, which we really don't need. So remove columns and genes metadata, and yeah, it's 3. We're gonna say start. We're going to say, I think it's end, and we're going to say width width. Yeah. Meanwhile, we are doing this, our output from replace text on data 3, it's ready, which if you remember is the count matrix, and we're just going to rename it as count matrix. Just so it's easier to remember later on our inputs. The next sanitation steps involve handling the samples metadata output file from the decoupler tool, and we will have to process through two tools this file, and it's important because the samples metadata file is the one that will help us generate the contrast file later. So we click on replace text. We're gonna choose where is it? Samples metadata. And again, we're going to ask the tool to find this pattern and to replace it with an underscore, just in case there's something there. And then we click Run Tool. We have to wait until that is done to use the output of that replace text into another sanitation step. In the meantime, we're going to name this genes metadata, gene metadata. Great, we have now our samples metadata ready. You know what? We're not going to change the name just yet. We're going to use now the replace text. In column 2, we're going to ask to find this pattern. You don't really need to know what this means, but you just trust me that it will help you handle the files easier later on, and to replace it with this. In detail in the tutorial, it will be explained here what the steps are doing for you. So in case something is not clear and you really want to edit it by yourself without strictly following the tutorial steps, it tells you here what it does. And yeah, great. We have now our samples metadata file ready. I'm just gonna name it as samples metadata so that we don't get lost later on. These 3 files are very important because they are needed for the EdgeR tool. But we need another file for the EdgeR tool, which is the generating of the contrast file step. What we will do is we're going to text reformatting, and automatically it will pick the recent acceptable format file, which is samples metadata, or tool, the text reformatting tool. Now we have here this awk program code. So with this cool code, you will be able to automatically generate a contrast file based on our samples metadata file. I would like to directly copy this code from here, but the problem is that if we do it like that, it will be structured differently. The code should not be written in different lines. It should just look like this, and not like in the tutorial. It is the same code, it's just here it is in different lines, and the tool for some reason just processes it not correctly, but we will see now. We will have our file generated. And here it is, our contrast file. We're gonna rename it as contrast file, And I just noticed that I've been explaining this tutorial without naming our history. We're gonna call it Pseudobulk Analysis. Okay, now we have our files. And here we have a question. How does your context file look like? Well, it has a header. It's important that the first row has this label, header, for the next tool. And it has the contrast automatically from our samples metadata file, which is bone marrow minus PBMCs. Our next step is to perform the differential expression analysis with our tool EdgeR. So EdgeR briefly is a commonly used tool for differential expression analysis in Pseudobulk, but also in bulk RNA-seq. And here we have an explanation of what the tool does and the statistical methods that it uses to calculate differential expression. And you can read it by yourself. But for now, we're just going to directly run EdgeR. So for our tool, we first have to input our count matrix, which is a single count, and we will find it in our history is the count matrix. Input factor information from file, we say yes. And the factor information, I believe, is the samples metadata. Genes annotation, yes. We have a gene metadata file. And here we have to write a formula. So I believe this is explained in the tool. We will go back to the tool in a bit. And it's also explained here in the tool, but it's also explained here in the tool section what the formula is and how it should be written. And it basically comprises this part plus the factor that you're trying to compare, which is tissues. You can input the contrast manually, and you will have to write it like something like this, or depending on if you want to group the factors or so on. In our case, we have it. It's called file, and it's a contrast file. So we have our information. Let's review it. We have the single count matrix. We have the samples metadata, the gene annotations, our formula, our contrast file. And in the section of filtering low counts, there's a part that is asking us to filter lowly expressed genes. And we want to say no. There's also more options here. We get to generate outputs for normalized counts tables, but we don't need it, and so on. And these other details are if you have more advanced knowledge in EdgeR and statistical methods. So this is just the standard, but there are reasons why one will use a likelihood ratio test versus a quasi-likelihood F-test and so on. As for now, with our data, we're just going to do it like this, and we're going to click Run Tool. And we will generate two outputs. One is a collection that will contain tables, and the other one is an HTML file with a report in which you will be able to visualize nicely a lot of plots and information from our differential expression analysis. The cool part is that if you click the data's output is not ready, but once it's ready, you will see here the option for saving it. Like, for example, here you will see the saving icon. And once you save it, you will be able to see individually these plots if you need them for adding it to a presentation or something. Okay, so we have our results. We have our collection first. This is a table. It's only one table with our results. And there's a lot of columns here. So it's not so friendly for visualizations. But in this table, if you download it, you will be able to see log fold changes and p-values. Now, Um, here's another file that is interesting, and is this HTML file that you can download. And as I mentioned earlier, you can individually as a PDF or just in an image file. You can get this MDS plots, this BCV plots, this QL plot, all these plots. And it gives you, in this case, a small information, like a brief summary of the differential expression count. So you will see that when we compare bone marrow to blood, we see that there are 188 differentially expressed genes that are upregulated in bone marrow, and they are downregulated in blood when compared to bone marrow. So if the contrast will be opposite, it will be blood minus bone marrow, then we will have 136 genes upregulated in blood and 188 down in blood compared to bone marrow. So it's just the opposite. The contrast, it just tells you the focus of this comparison. And here we have also the groups that we analyze, the names of our samples, and so on. And the expressions, the log-fold changes, and the average expressions, how much the fold change it has. And maybe these genes are the ones that are the most interesting, and we will need to label them in order to see what's the name of them, or just look at the table directly. But since I already showed you, the table is very big, so the best thing to do will be to just crop the columns that are interesting. Now here we have a couple of questions. Is what are the outputs of the EdgeR tool? As we already saw, we have EdgeR tables and the important information that it contains. We have as well the EdgeR report and what it contains. And what do they mean as well? So you get to read here what is the interpretation of them, which I already briefly mentioned. The other question is, about interpretation. How can we interpret our output results files? And I already did this with you with the HTML file of the output of the EdgeR file. Now the next step is the sanitation step, and is the sanitation step part 2, which is pretty quick. And as I mentioned, the table is output from EdgeR as a collection, and we cannot directly run sometimes important tools or needed tools, like if we want to create a volcano plot with our tables, just by selecting the columns of this table that are needed for the volcano plot. We cannot do it if the table is in a collection. So what we're gonna do is first we're gonna extract the data set, and since it's only one, we don't need to change anything here. It's just a table from EdgeR output, and it will extract the first data set from my collection. And it's already here. It's nothing, not a big work for the tool. The next step is to remove columns. So we're going to use this tool called Remove Columns to keep only 4 columns from our tabular file that will be useful for generating a volcano plot and visualizing in a better manner the differentially expressed genes. So as here it says, the columns that we need to keep are the X, the log fold change, the p-value, and the false discovery rate. The X is, in our case, the column that contains the gene names, but in a different data set, depending on how the annotation or the metadata is labeled. This header will have a different name. It could be gene ID, it could be gene names, or it could be written differently. So you have to put there the header that corresponds. The other header that we will keep is a log fold change. There is another one, which is the p-value, as I mentioned. This is the raw p-value column. And the fourth one is the false discovery rate, which is the adjusted p-value. This is important. We must say, yes, we want to keep this name column. So the ones that I selected here are the columns that I'm going to keep. And we're going to copy this already because the output file, we will rename it like EdgeR DEG summary. We have now our summary from the EdgeR tabular file of the differential gene expressions. And this is easier to read. So here we have our column with the gene names, the log-fold change, the p-value, and the false discovery rate, which is the adjusted p-value column. Now, the next step is to generate this volcano plot. And this will allow us to visualize the relationship between the p-value, which with a statistical significance, and the log-fold change of the differentially expressed genes. And for that, we needed that file with the 4 columns. So we're just going to directly get into it. I think, yes, the file has a header. And the thing that we will do in this tool is we just input the column that contains this information. So FDR is the 4th, this is the 3rd, the log-fold change is the 2nd, and the column with the, this is the column number, labels, the gene names. Significance threshold, yes, the ones that have an 0.05 p-value, and points that will be colored are the ones that are significant. And in this case, let's just say I think it's 40. We're going to label the topmost significance. And in plot title, we can also make adjustments accordingly because you can generate plots here for publications as well. You can say label boxes and so on, but we are just going to name it like that. And you can adjust this and customize it depending on your needs, right? But that's about it. That's all we need. So we click Run Tool. And the last step should show us a volcano plot with our differentially expressed genes. And this plot will show us the genes that are up-regulated and down-regulated that we saw in the analysis results. So in this case, we know that the bone marrow has 188 up-regulated genes when compared to blood, and has 136 genes down-regulated when compared to blood. So always remember that that is the comparison that we are doing based on this contrast, and that's what it means. The genes that are up-regulated in bone marrow, and the genes that are down are also down-regulated in bone marrow compared to blood, all compared to blood. If we would have chosen an opposite contrast, then it would be 136 up in blood and 188 down in blood when compared to bone marrow. Now, we're just going to wait until the job is done. And our job is ready, and we have our volcano plot. We can visualize it. Here's our title that we can customize. And here are all the genes that are down in bone marrow and that are up regulated in bone marrow when compared to blood. So as we see overall, our cells, our non-classical monocyte cells and our dendritic cells express different genes depending on the compartment they are in whether they're in the blood or in the bone marrow. Now, Yes, here we have a tip and the results. We already discussed them. Here are some questions, and you can also read in detail later on. But for the last part of this tutorial, we are going to be subsetting samples from the original and data if you want to run this differential expression analysis in each of the clusters that you found in your single cell data. So when one performs single cell data analysis, we end up with different number of clusters and different cell types, because that's the purpose also of single cell, of identifying clusters and annotating the cell types. In our case, in our data, we have non-classical monocytes and we have dendritic cells. So let's say I want to see if only non-classical monocytes have genes differentially expressed or express different genes in the blood versus the bone marrow, but without including also the dendritic cells. Or let's say I have these results from my Pseudobulk, right? In total, I mean, I had 3 bone marrow samples from, from non-classical monocytes and 3 bone marrow samples from dendritic cells, and then I have 7 from blood from non-classical and 7 from dendritic cells. In total, I had 14 blood samples in my previous analysis and 6 bone marrow samples. But if I want to run the same analysis within one cluster only, then I will only have 3 versus 7, right? And we will have to subset our clusters from our AND data in order to run this analysis again. And for example, in this case, we have a question. I know that there's a difference in the expression of the genes of these immune cells when they are in bone marrow versus when they're in blood. But is this difference really driven by a specific cell type? Is it like heavily that is one cell type that is driving this difference of expression in different tissue compartments, or are they both driving these differences? And this is what we can answer by subsetting. We get more information. But in this step, I'm just going to show you how to subset with a scan by filter tool your clusters. So there's a way to do it one by one, but if you want to subset directly all the clusters, there's another tool that is called manipulate on data, and you can directly generate a collection with all your clusters, which is faster. But if you're interested just in one cluster and you just want to extract that one cluster from your data, we can totally just use scan by filter tool, which is what we will do now. Here we have our annotated data set. We're going to filter by observations and variables. We're going to filter observations because we want our cells. Key to filter. It's annotated in our case because that's under annotated is where the names of all of our clusters are. We say text because equal to, I believe the name is, in the first scenario is PDC, which is dendritic cells. If we want to run this Pseudobulk analysis again, only in dendritic cells. Now, it is also said here that you can also just put the name of non-classical monocytes, and then you will be able to filter only this cluster and run the same analysis on it. So, yeah. These are advanced options. We don't need it. Hit run, and we should have our data with only the plasmacytoid dendritic cells. Great. Our file's ready. And now we see here that we only have the dendritic cells, which are 1092, and of course the whole set of genes that are expressed, and then we will know whether they are in one cell expressed or not, and so on. But this is data extracted only of this cluster. And we can do the same for the monocyte cluster. You just have to make sure to put the right name of your annotated cluster in the tool. It has to be here the right name that it's in the data as it is. Okay, so let's say we perform the same steps after extracting this. We just go back to square one here, and we run the decoupler tool and all the other steps only on this plasmacytoid dendritic cell cluster. Let's just label it like this. We have this cluster here. We can do the same for the non-classical monocytes. Now, here are some questions. What data is included in the new Pseudobulk count metrics for the plasmacytoid dendritic cells only, and how is it structured? What do column labels represent? How many samples are included when you run the Pseudobulk, and after performing on each individual cluster independently, how do the volcano plots compare? Do they reveal differentially expressed genes between tissues? If so, which cell type appears to drive the tissue-specific differences that we were talking about? So, yeah, so this is strictly after we have run our Pseudobulk and generation with the coupler. We will only have 1,373 rows representing genes and so on, and yeah, we will only have this amount of genes. In any case, you will have to perform this separately. But in the answer history, if you go back to here, you see that we have 3 answers histories. One of them is the answer history for the plasmacytoid dendritic cells. And here we'll have already all the plots. And before I continue, also, if you don't want to extract it and you just want to run it in the workflow directly, this cluster, you can just directly download these datasets from Zenodo by clicking download here. In any case, when performing this bone marrow versus blood comparison in the plasmacytoid dendritic cells, we see here in the volcano plot that there are no significantly differentially expressed genes. However, when we perform the same analysis only in the non-classical monocytes, we see that there's a lot of up and down regulated genes in bone marrow. This means that the main contributor to the tissue-specific expression patterns observed in the full dataset are the non-classical monocytes. So yeah, this is it for the Pseudobulk tutorial. Thank you for going through it with me. I hope that you learned something. I hope that this was clear. If not, please feel free to contact us and to send your thoughts or to ask for help to the Galaxy EU community. We're always happy to address your doubts and your concerns. And here are some recommendations of the data preprocessing, the focus, the quality control, what other analysis you can do with this differentially expressed genes data. A type of analysis that is very interesting is the GO enrichment pathway analysis after you have generated the differentially expressed genes. And yeah, future applications. So you finished this tutorial. Congrats. Thanks so much for sticking through all the way, and I hope this was useful. Thanks very much.